in this. Okay, so for future predictions, I pulled up this chart together where I'm trying to indicate where the global bioinformatics market is, because it's very easy to talk about academia and bioinformatics, which is great, of course, and it's there on the university. So I'm going to target basically the industrial aspect of it, and then slowly get down to the academic part. So as you can see in this chart, very well, like global bioinformatics market between 2018 to this year is everywhere, like in the rest of the world, including Asia Pacific market is of the regional medium, the Europe market is of very high, bioinformatics market is there, and Northern America, which is basically comprising of United States, has a very, very high market for bioinformatics, knowledge, bioinformatics, resources, bioinformatics tools, bioinformatics services. So in this whole everything that has been going on, India has become the first country to establish a nationwide bioinformatics system, which is under the body of ICIASTSF. It was started in 1986 by the Now, bioinformatics has attracted several companies and entrepreneurs. This market has been persistent. Under the recession came, overcame, but this market still exists. The bioinformaticians who were working in industry or academia, the lab was not shut even for a few. So this is a complete, this is a revenue that has been everlasting and has been going on without any break, without any action. When the world stops, bioinformatics still keeps going. So that's a very good thing. Now, the global bioinformatics market has grown from 2016 till this date. I mean, it's a future prediction. It has grown from 25%. If you match these stats with any of the other industries, market or any other thing out there, you would see that 25% of growth is too much. It's a very significant thing. There has been no downfall all of this whatsoever. And these stats are now, with this, these bioinformatics approaches, they all analyze several data that has been provided from different resources. So I've tried to sum up that where is all this data coming from globally. We know that research data comes from labs, and sometimes there are like genomic sequences that come, but in order to get a global approach towards it, I'm just trying to sum it up here. Now, there are three main entities that generate this data. One is the GenBank, the second one is EMBL, and the third one is DDBJ. Now, GenBank and EMBL, GenBank is in the United States of America, EMBL is in Europe, Heidelberg to be specific, and DDBJ is in Japan. Now, these three bodies are the central bodies that keep exchanging the data within itself, and this data could be anything from genomic sequence to proteomic sequence, RNA-seq, tag-seq, you name it. This is the channel it goes through. Everything available in the name of bioinformatics databases goes and gets certified or clarified from them. That's where all this global data is coming from. Any researcher who's doing any wet lab and is using any nanospore technology or any other sequencing machine, this is where the data finally gets at and gets deposited, right? So this is the main resources that you would get your data from. And it keeps getting updated every 60 days, all three of them. Now, in India, we have the Biotechnology Information System Network by the Department of Biotechnology, Government of India, and they have been working towards many achievements and trying to advance bioinformatics in the country, because I'm going to focus my talk today in Indian context mostly. So they aimed these projects to provide national bioinformatics network designed to bridge the interdisciplinary gaps in biotechnology and establish a link among scientists and organizations involved in R&D and manufacturing activities in the industry as well. They have also tried to build up an information resource, prepared databases in biotechnology that could be accessible to each and every academician or industry personnel. And they continuously access information requirements. They update it and keep it accessible to everybody out there. They coordinate their efforts within the system, and along with that, they also coordinate their data with the global databases on genetic material, their published literature, and any other scientific and commercially valuable asset that is related to bioinformatics. They perform search into advanced methods of computer-based information processing, which is definitely comes under the aspect of bioinformatics, and to evolve and implement an education system for users and training at the same time to bring the wet lab researchers up to date and be able to access this. Moving along, the BTIASnet that I mentioned a few minutes ago, it has shown great promise, and it has an outcome in last 3 years. It has made publications of over 1,280 publications. It has the patents and granted, filed and granted patents of 56. And the number of manpower they have trained over the years is 8,998. The major facilities and programs supported by BTIASnet are Indian Biological Data Center, IBDC, the Biotechnology Information System Network, Manav Project, the Human Atlas Initiative, Indo-Japan Diacentric Program, DBT Apex BTIC, and Northwestern Himalayan Bioinformatics Centers. So I just tried to put together this map of India indicating the several places where these centers are. So again, I'll be sharing my slides. So please go ahead and take a cursor look if you want to. But these, like the captions are here, the center of excellence, like the entire country is covered. We may think that bioinformatics is new, but it's not. Like the entire country is covered with bioinformatics centers and small initiatives, big initiatives, and so on and so forth. Moving along, let's talk about the Indian Biological Data Center, which is in pilot phase now. This basically involves the agricultural data sets, livestock data sets, microbes, and human data sets as well. Now, this involves the deposition of biological data, their quality control, and curation, annotation, distribution of data. And at the same time, they also create the data backups, management, and they provide trainings and outreach activities as well. This initially was to access the modality and accessing the amount of data generated and developing methodologies. And all of these, they provide a specific training via DBT and the Government of India. There are like several trainings going on every year, and actually every 3 months, I would say. I was fortunate enough to take one of these trainings while performing, like while doing my PhD with ICGBI and ICMR. So these opportunities are definitely there. Just go to these websites and there'll be a lot of trainings that one could take up and take help with. Now, another thing which is really taking over the world right now, will not even say that just India, but the world that has been taken over by is the artificial intelligence and bioinformatics. Now, here I'm trying to represent an image of biobank for cancer. As I said, India is working in all forms, agriculture, poultry, microbes, and humans. So when we talk about human, cancer has been a big data generation has taken place. So this data bank that has been created, the image bank store, it creates the information from radiology, radiomics, annotations, their labels, digital pathology, and this is a small follow-up. This chart is a small follow-up how to annotate what kind of process it goes through. So they have the data from diagnosis to planned events, then response assessment, follow-ups, and so on and so forth. So as you can see, from the day of diagnosis till the person is cured and sent back to their family, everything has been annotated and has been curated with the help of artificial intelligence in the imaging biobank for cancer. Now, this, I think I already covered this, but this project is being supported by Tata Memorial Center, Advanced Center for Treatment, Research and Education in Cancer in Mumbai, All India Institute of Medical Sciences, AIIMS, Delhi, Indian Institute of Technology, IIT Bombay, and Rajiv Gandhi Cancer Institute and Research Center at the unit of Interpers Cancer Research Society, Delhi. So these bodies are distributed all across India, and they collaborate. Even though they are like miles apart, they work together. And that's the beauty of having bioinformatics and artificial intelligence, is that's how they are getting the human cognitive system to work. Now, what can this data be used? We understood that these data has been taken and it has been curated and they are doing follow-ups and everything. But to understand how this data can be used, I've tried to put together this slide. So first and foremost, there could be predictive tools. If you take the data set from a patient and try to train it on a new data set, we could identify genes and targets which would cause a certain kind of cancer, myeloma patient, breast cancer, triple negative breast cancer, colon cancer, everything. So these predictive tools can be developed using this data. Then there could be, with the help of these predictive tools, there could be an automated diagnosis. Everybody can have, with just a swab, you can predict what kind of cancer you may be prone to later on in your life. So all these predictive tools and all these predictive automated diagnosis can be developed using artificial intelligence with all this data that has been coming through these bioinformatics approaches. Now, there could be a lot of questions when somebody gets into into a sick phase. They have a lot of questions about how it's going to hamper their other aspect of life. All those, and there are research questions too. So all those questions could potentially be answered. There could be disease-specific questions. There could be prognostic tool-related questions, or any sort of questions. With artificial intelligence, we are more capable of answering those questions, whether it's health-wise or research-wise. Now, the collaborators for these, I've put together the logos. So wherever you see these logos, just understand that they are working in artificial intelligence and bioinformatics for the country. So we should be very proud. Now, the another initiative by the government of India is MANAV, the Human Atlas Initiative. I really like the name because it's Atlas. Atlas is a word that defines or indicates a global context. Like whenever we say Atlas, we think of the map of the world. So what they have tried to do here is Human Atlas Initiative is they have tried to have a holistic approach towards bioinformatics, where I've tried to put this image together where you can see the key components are literature and database annotation, then data validation and collation, then identifying gaps and missing links, and the community development and collaboration. So as you can see, with the MANAV project, they have kind of captured all of it. So like from data generation to the curation, validation, collation, and even getting it till the community development and collaboration level. So that's a holistic approach, and which is why the name Human Initiative Project MANAV. So the link is obviously there on the slide. Please make some time to go ahead and explore these resources that are there by the government of India and DPT specifically. Moving on, the MANAV, the Human Atlas Project, it is a collaboration between ICIASTSF Pune, Persistence System, and the National Center for Cell Sciences, and it was launched on May 10th in 2019 by the DPT Secretary Dr. Venu Swarup. It has 5 million scientific articles. It has many students, scientists, research fellows. There are research master projects, and everything else is, like, as I said, it's a holistic approach towards having biological data and bioinformation put together. Now, the last but not the least, the Indo-Japan Dicentric Program. Now, the Department of Biotechnology and the National Institute for Advanced Research, IST Japan, has initiated this program, DBT, IST, and this was established as IST Japan Tsukuba campus and conducting advanced biomedicine translation and environmental research in collaboration with Indian Research Institute, IT Delhi, and RCB Faridabad, Sikkim University in Sikkim, and Guwahati, of course. So these are the projects. I'm just telling the names and the collaborative projects that they have put together, and of course, you can explore the website. They have more information there. The next one is the DELCON, which is the DBT's e-library consortium. So it is a summation of getting all the literature related to bioinformatics and artificial intelligence or any other data set like that put together. And it is a significant initiative by DBT, of course, to enhance the information resources in its research institute. So it's more research institute based. So students and scientists and PIs can have more access to all this data that are generated through all these previously explained bodies. And it was launched in the year 2009, January, with 10 DBT member institutions with a large number of prominent handpicked online journals. And till this day, they have over 1 million literature deposited in their corner. Now, you may be thinking that I'm all talking about bioinformatics and everything. What is the future? Like, the whole idea of today's conference is like, what is the future? So some people ask, like, what is the pay scale student would look up to if they are taking bioinformatics as a career, or what's the future in it? So the pay scale in bioinformatics field is dependent, of course, on the position that you're working on and as an individual as well. But initially, bioinformatician is expected to go anywhere around 2 to 2.5 lakhs per annum. And as experience increases, the salary increases accordingly, whether you are in academia or you are in industry. And then, I mean, these numbers are all good and great, but I would say sky is the limit. And these stats are for India. These are not for abroad. If you work in collaboration with abroad, these stats go even higher. Now, the other question I get a lot of times is bioinformatics a viable career? Like, like biotechnology came in after 10 years, everybody was a bioinformatician. MBA came in, and then we have MBA graduates everywhere. So is bioinformatics a viable career? I would say, of course, it depends on the individual, but bioinformatics is something which is an everlasting, and it's kind of ever-growing, dynamic kind of system. So there is always going to be data being generated in labs. There will always be a need of analysis. There will always be a need of tools to access and analyze those data sets. So bioinformatics is something that is ever evolving. So this is definitely a viable career. The only thing is you have to be dynamic and keep growing with the system. It's not a stagnant system. That's the only thing. So if you're looking for something that, okay, I'm going to learn one thing and just sit back and have a paycheck to paycheck, you may want to reconsider being in bioinformatics. So it's an everlasting and ever-growing and ever-changing field, but it's never going to die out, of course. It's going to change from one form to another. Now, what is the future of bioinformatics? I think I've given enough of talk about the future of bioinformatics, but it's expected to grow and integrate itself in more efficient areas outside of research. Services like hospitals, general practices, services direct to public are some of the examples, of course. Medical billing is a new term these days that has been popping everywhere in India and abroad. Not a lot of people say this, but even medical billing is a form of bioinformatics, because there is a system being created and you're putting in information like a bioinformatician, you need to have the uh, bioinformatics preliminary knowledge in order to do that. So that's where the future of bioinformatics is headed in terms of science and technology, and it both ways, both in academia, both in industry. You can have government positions and government work while being in bioinformatics, and you can have industry and your, like angel investors. You can have your company, you can have foreign investors. I mean, the sky is the limit. You just have to look and work for it. These are some of the references I used for putting up the slide together. And with this, thank you so much for your patience listening, and I'm open for any of the questions that you may have.
Yeah, madam, it is very informative and very interesting talk. Anybody have any question? Let me ask. Right now, CRISPR is a new technology because based on this technology, and really, ma'am, we are very proud that we have so many resources that the government of India has given to us that we will be able to complete gathering. So we are very proud of this, and definitely it would help in the upcoming technology that is CRISPR-2, and genes which are involved in the cancer and other diseases that may also be directly targeted by this technology. So thank you so much, ma'am.
My pleasure. Thank you so much for the opportunity.
Over to Rahul sir. Thank you so much, Dr. Kalyani Vidya ma'am, on this research. Sir, I have one question. Yes. And, how we can explore the molecular docking in phytometric space?
Just for phytomedicine?
Also, ma'am, broadly we can say medicines. What kind of this? How we can use some phytomolecules or some secondary metabolites in analyzing the molecular docking and gene expression level, and how the bioinformatics can be efficiently utilized in molecular docking system.
Right. Thank you for your question. I can't see your name, but Kumar sir. Okay. Okay. Well, thank you so much for that question. There's one thing, like, yes, I feel like you have put together multiple things: molecular docking, gene analysis, and everything else related to phytomedicine, which is great because now you're exploring. So you're halfway there. So congratulations. Now, with phytomedicine, how it would work is, let me try to answer this in bullet points. With molecular docking, if you have any phytomedicine, or if you have any ligand, I would like to call, and if you have the chemical formula for that, there are tools that you can draw the structure of. You can draw these structure and you can save them in PDB files or STM files. These are the file formats required in bioinformatics, and you can use that into a system called QSAR, where you take this structure and then you can predict the target for several uses. If you're looking for a specific pathway, then you can even utilize that. If you're looking for a specific disease related to those phytomedicines, you can target that as well. Because there are databases for pathway channels too. There is KEGG, there is SPRING. other thing that you asked me is about how could we do like gene analysis and everything. Now, first question that needs to be answered is, are these phytomedicines in any way interacting with genes? How do they change the expression of genes? Now, if that question is answered by literature that they do affect certain kinds of genes, then definitely there is protocol in bioinformatics to approach that as well. The resources that I would recommend for you to follow up and go through is just go to ICAR's website, and they have a separate bioinformatics section called Kevin. It's a very fancy name, CABIN. They have trainings every month, I guess. Like, they're very proactive. They have a bunch of young scientists there, PhD students there, and they have trainings too. And if not, there is like online resources. With COVID, the online resources are very popular. We have Udemy, we have Coursera. If you have an institutional ID, just register for free, and I think they give a subscription of 6 months. It's all good and great. For more queries, please do write to me. I'll be happy to discuss this with you.
Thank you so much, ma'am, for your kind enlightenment. Thank you so much.
You're welcome. Sure. Thank you so much for having me. Have a great day. Bye-bye.
Next speaker is Dr. Pramod Ramteke, sir. Are you there in the meeting? Dr. Pramod Ramteke, sir.
Yes, yes.
Good
Hello.
morning. Good morning, sir. Good
Good
morning.
morning. Good morning. Good morning.
Good morning, sir. So, dear participants, our next speaker is Dr. Pramod Ramteke, sir, ex-dean postgraduate studies of FH University of Agriculture and Technology and Science, Prayagraj, Uttar Pradesh. And sir will be introduced by Dr. Subhash R. Sompur, sir.
Okay.
So, over to you, Dr. Subhash Sompur, sir.
Thank you, Dr. Sir. May I be audible to all of you?
Yes, sir. Yes, sir. You are audible.
Okay, sir. Thank you, sir. It is my proud privilege and a great honor to introduce before this scientific gathering, Dr. Pramod Ramteke, sir. Professor Pramod Ramteke, sir.
Yes. Hello.
Okay.
Can you hear me?
National Academy of Biological Sciences, Mycological Society of India, and International Society of Environmental Botanics. In view of his great contribution in the field of for that, various awards and honors were conferred on him, like Banaras Hindu University Centennial Award, Excellence in Science Award, J.C. Bose Gold Medal Award, Dr. J.C. Edward Medal Award, Professor K.S. Gilgrani Memorial Award, Professor K.V. Shastri Gold Medal Award, P.S. Chavan Gold Medal. List is so long. So, n number of awards he received because of the great contribution in the postdoctoral fellow under his able guidance, and 4 patents he received, 2 technologies he designed and developed, 5 databases, and 323 gene sequences in National Chemical Bioinformatics he submitted to his credit. He authored and edited 10 books and over 250 research articles, including 6,308 citation index, as per the recent Google. He served as a. Am I audible?
Yes, sir. Yes, sir. You are audible.
He served as a referee for reviewing scientific articles in various reputed journals such as Journal of Flyers, ACS, elsewhere, Spingers, Wiley, Taylor and Francis, and so on. He is nominated as a subject expert and chairman, state and national level environmental and animal ethical committees by Ministry of Environment, Forest and Climate Change, Department of Science and Technology, Indian Council of Agriculture Research, Indian Council of Medical Research, and Council of Scientific and Leadership Research, Government of India. He was a visiting scientist to Hungary, USA, UK, Belgium, Turkey, Czech Republic, and South Korea, and member of 18th Indian scientific expedition to the Antarctica. With these brief introductions, I would say thank you so much to Professor Ramteke sir for taking initiative to organize this international scientific mega event. Because of you, this event could possible. I am very much thankful to you, sir. And with this brief introduction, I request you to please enlighten this scientific gathering. Thank you so much.
Hello, good morning. Can you hear?
Yes, sir. Yes, sir. Your voice is very much clear.
Good morning, Dr. Somkumar and Chairman of the station, and report here, and all the delegates and members who have joined us this morning. Indeed, it's a great pleasure for me to be with you this morning and share some of the ideas, concept, and also some of my work with you, so that we all will be benefited and continue to work on a sustainable development, and especially sustainable agriculture. Yeah. And ये जाएगा. Right. Hello. Can you see the screen?
Yes, sir. Yes, sir.
Can you see this? Okay. All right. So today I will just want to discuss on microbial diversity in sustainable agriculture development. See, we have development in India. And we had many high-yielding varieties of wheat, rice, and other grains were introduced in India. And we increased our production of crops. And so India became a self-sufficient in food grains. Create more jobs in agricultural laborers, and also we have indirect benefit for industry. Okay, we could have many small industries and many other industries, both in rural as well as urban. And country could go from importing food to exporting it. So not only we were importing food, but because of regulation, we could export it. And because of all this, we have a major social and ecological impact. So there were no more famines after the revolution, and the revolution has transformed our country from being bowl to bowl full of rice. So now we have come to the optimum of the revolution. Okay, we have to stable. So there is now stagnation. Because of this revolution, we also have some adverse effects. We started using a lot of synthetic fertilizers, a lot of herbicides, pesticides, and a lot of irrigation. And this has created a number of problems for us. See, now our insecticide consumption raised 76%, herbicides 10, fungicide, and all others. So this is a major effect on our soil. So we have witnessed environmental pollution, soil degradation, eutrophication, and their health hazards has been also reported. So these are the adverse effects of our green evolution. Now, our population is increasing. Okay. So how we are going to cater to the needs of our food? And we have a big challenge. Dr. Ramteke, sir, you are not audible, sir. Yes, sir. You are not audible, sir. Honorable Pramod Ramteke, sir, you are not audible. If there is any technical problem, please mute the meet and please rejoin. So that we can hear you. Chairman, what problem is going on? Chairman of this technical section.
Hello. Hello. Hello, sir. Is there any problem? Hello, Ramteke sir. Are you there in the meeting? Hello, Ramteke sir. Hello, Ramteke sir. Hello. शायद रामटेके सर का कनेक्शन डिस्कनेक्ट हो गया. It's not in the meeting.
Yeah. I think we should call him and tell him to rejoin the meet.
Yes, sir. Yes, sir. शायद.
Do you have a number, phone number, sir?
No, sir.
Okay. Right now I call him.
हाँ, आप कॉल कर दीजिए.
Have we joined?
Hello, Ramteke sir. Yes,
Now it's
sir, I'm
okay?
fine. Yes, sir.
Okay. Yeah.
Yes, sir. Sir, please share your screen.
Yeah.
Yes, sir, it is visible.
Right. Sorry for interruption, sir.
No, sir, it's okay.
All right. So microorganisms plays a very important role in agriculture, and they have multiple modes of action, both for growth, defense, and health. Let us see one by one. We produce a lot of hormones. They produce antibiotics. They can mobilize nutrients. They produce hydrophores. We can get iron and other nutrients. Produce chitinases. They produce deaminase. One by one, we are going to see all of their action. They produce SCN. They can fix atmospheric nitrogen. They can do the nutrient mineralization. They can do the pesticide degradation, and also they can induce systematic resistance. So these are all their mode of actions which are helpful for growth, defense, and health. So this is an overall chart, both showing direct plant growth promotion and how they help in a indirect way, especially taking control of their health by their biopesticide activity, which we have already seen. This is a summarize of that. Very important thing we know is that they have ability to fix the atmospheric nitrogen. We know that we can reduce a lot of nitrogen load by external use if we can use this. Nitrogen gas, it turns into ammonia, goes into the legumes, and legumes can fix this nitrogen and again supply to the plant. Siderophores are also very important for iron intake. Okay, they chelate iron. Iron is very essential nutrient for a plant. So this is the entire process of siderophore production and how they can chelate the iron. Another very important mode of action is ethylene is. Okay, we all know that ethylene is the large production of ethylene is very harmful to the plant. So this enzyme breaks down the ethylene molecule into ammonia and alpha ketoglutarate. And you know that ammonia can again recycle for production of fertilizer, and alpha ketoglutarate is the precursor of hormone, especially indolacetic acid. So by breaking the ethylene, we have two important products which will help for a plant to fix the nitrogen and also to produce the hormone, indolacetic acid. We also produce a lot of phytohormones. Okay. And these phytohormones, especially indolacetic acid, can take up in the plant, and then they produce the hydrogen molecules. Where by that the turgor pressure develops and many of the proteins are activated. So this is a mechanism of phytohormones. Okay, there are several number of compounds which acts like antibiotics. That means they stop the growth of pathogens, like SCN. It controls the weed. Phenazines control the fusarium, oxysporium. Chloroglucinol controls the pythenium. Pyrrolnitrine, rhizoctonia, and there are polymyxin and polystine, which also control the gram-positive and gram-negative bacteria, and then bacteriocins, okay, which stop the entry of other pathogens. So these are all some of the production of antibiotics by the plant-growth-promoting bacteria. Plant-growth-promoting bacteria also utilize pesticides as a sole carbon source. So now, whatever pesticides we use, especially organochlorine, carbendazim, and carbofuran. Okay, these are some of the organisms which they degrade. Degrade means basically they use this compound as their basic carbon source. They utilize this compound as a basic carbon source. The heavy metals also, they take a very important role, especially there is a process of adsorption, then absorption, and some of the these microorganisms has enzymes to transform toxic heavy metals into process of detoxification. Like chromium 4 is a very toxic, so they produce the zero valency chromium. Okay, mercury also very toxic, so they produce gaseous form of mercury, gaseous form of chromium, so that there is a process of detoxification. They play a very important role in nutrient mobilization, especially we know that we need a lot of phosphate. Okay, so this bacteria has ability to solubilize the phosphate. See, a lot of phosphate we cannot take it right in the soil. So what they produce the organic acids, which solubilize this phosphorus and also provide to the plant. So phosphate solubilizes bacteria plays a very important role. So this is a whole process of nutrient mineralization, okay, from dead and decay matter, from animals, okay, from other things where they by their metabolic activities they can degrade or they mineralize they from complex form to simple forms. They turn these important nutrients and make it available to the plant. Okay. They also produce a lot of polysaccharides, which improve the soil aggregation. Now we have a lot of problem of soil erosion. So these polysaccharides they bind soil particles together and help soil from the erosion. Okay, so the erosion can be controlled by microbial polysaccharides. So plants are, because of over a lot of pollution and soil degradation, they are stressed and they are trying for help. Okay, and in this case, this root associated bacteria and fungi, they come forward. They say, okay, we take care of. So they help in management of all kinds of stresses. Okay, BCPR, they produce cytokines, so they take care of ROS and AVA in a broad. We have a salinity problem, so it's CTA minus, blocks the insulin and also take care of salt problem, and then this nucleoacetic acid also helps to provide hormones to the plant. So in all the traces faced by plant in growth, defense, and health, this BCPR helps. So as we said, this is a like summarize both biocontrol of plant pathogen and plant growth promotion. This all this is different mechanism what we have seen, and now they provide both the benefits. We have conducted some work in Allahabad, now Prayagraj, and we isolated around 650 microorganisms from organic farm, and we got very useful organisms, both production of ammonia, SCN, hydrophore, indolacic acid, SCCD minus, phosphorylation, and catalyst. And large number of organisms are available. We also studied their salt tolerance, and we found most of our organisms are tolerant to 5% of salt, some to 10, but there are few organisms which was tolerant to very high salt concentration. It was something very surprising, and we repeated this experiment 3-4 times so that we are sure that what we are telling is okay. So there are 2 organisms from coliform and 1 azotobacter, which were very tolerant to high concentration of this. Then we also studied them for a pH tolerance, and we could get that acidic as well as alkaline pH. So these organisms have a broad range of pH tolerance. So in all traces, we can use this organism. All right. This the Bacillus thuringiensis we isolated from Allahabad region, okay, and we studied against these pathogens, and there is a lot of mortality after 72 hours. So for pathogenic control also, we had a collection of Bacillus thuringiensis. These are some of the experiments where millets, rice, maize, we put them for a growth enhancement, challenging with our isolates, and you can see a lot of growth, and as compared to control. These are another experiments. Okay, did this experiment, and this was a field, both laboratory as well as field studies, and you can see the real effect when we compare with the control. This is experiment with chickpea and broccoli, and you can see there is a large root formation, long root formation, and also shoot formation. This was with the maize and under the salinity. So I wanted to, we had some publications, and I wanted to group them in different categories. And when we come to plant pathogens and biocontrols, we have these papers. One came in scientific reports, and then another in a genome announcement. Then biological agriculture and horticulture, and National Academy of Science Letters, which is Springer Plus, and World Journal of Microbiology and Biotechnology. This is other with the suppression of bacterial wilt of tomato by plant growth promoting rhizobacteria, and we also did a lot of diversity assessment of antagonistic trichoderma. Mesorhizobium also we had a collection, and we challenged them against the phytopathogenic fungi, Fusarium oxysporum, and we could get good results. And both in chickpea. Then we also work on the metagenomics, and we did the wheat rhizosphere from 3 places: Balia, Gazipur, and Mau. And this whole metagenomic metagenomic is deposited in a LCPI, and these are the axis number. And we had a one good publication in data in brief of Gazipur region, rhizosphere, wheat rhizosphere of Gazipur region. This is another review came in the next generation sequencing approaches, discovering hidden microbial pressure in soil, and then another good chapter. Then we had some work on a plant growth promoting rhizobacteria direct. It helps the plant, and this is one review published in Microorganism, another review, and then these are our actual work what we did. These are different organisms, the heavy metal tolerant bacillus, and then heavy metal tolerant pseudomonas, and then this organism from Pakistan, and then we got rhizobium isolates. We also compared ACC deaminase production in selected plant growth promoting rhizobacteria. We did some phosphate solubilizing activity of endophytes in sugarcane plants, and solubilization of potassium and phosphorus, and use of these organisms in production of broccoli. We also did some formulations using halotolerant phosphosolubilizing enterobacterial protein and halotolerant. This is a paper, Journal of Control release, and we did on a sidrophore, and then on the fungi, in Journal of Fungi. So was one book published on fungal sidrophores, and some review article in Biotech Today. And then these are the actual field work we have. Okay, we use one of our enterobacter for growth of rice and millets under salinity stress, and millet under other abiotic stresses. This is tomato promotion. This is down one is the Irulinia. This is then the wheat under clear stress, and then under heavy metal. Yield of wheat variety. This variety was is developed in our institute, so we wanted to take this variety to reduce the fertilizer load and see how our native bacterium can work. And then we also use some photosynthetic bacteria. This is the growth of tomato using Azotobacter and Azospirillum, and seed germination of rice variety Shubhangi by Andrew Poeki. And we also had a paper on molecular aspects of symbiotic association between legumes and rhizobia. We also studied their antibiotic resistance. So microbes are very critically important, I call them. And Louis Pasteur says that the role of the infinite small is infinitely large. So they are very, very important. I want to acknowledge all my PhD students who have worked on this, all these aspects of plant growth promoter, rhizobacteria, Alka Sagar, Yogendra Singh, Ram Prakash Pandey, Govind, Priti Rathore, Supriya Kumari, and Dr. Mohammed Hafiz. I sincerely thank Professor Subhash Somkuwar and all the members of the organizing committee for giving me this opportunity to present my small talk. And thank you very much. If you have any things wanted to ask, then I will be happy to answer some of the questions. Is it okay? Hello.
Hello, Dr. Somkuwar, sir. Are you there?
Yes. Yeah.
Dr. Somkuwar, sir. Yes, sir, it is okay. Nice presentation, sir. If anyone is having any question, you may ask to sir. From the audience. Sir, I believe that there is no question from the audience side or even in the chat box too. Somkuwar, sir, are you there? Dr. Somkuwar, sir. Thank you so much, Dr. Pramod Ramteke, sir, for being with us and for sharing your valuable and very informative knowledge with us. I believe that definitely this knowledge will motivate us to move further in this field. So, on behalf of all organizing team, I would like to express my deep, sincere and thanks towards you, sir. Thank you so much, sir, for being with us.
Thank you very much. Well, sir, and thank you, Ramteke, sir. I have one question, sir, because of
Yes,
I'm in
please.
traveling. So you wrote one book on siderophores, and recently, as our Vidarbha soil is iron-rich soil, so in that context, what research we can do for Vidarbhian farmers so that we can at least contribute to minimize the farmers to the suicide cases in our region.
Yeah. Thank you for this very important question. Yes, we have our soil is rich. Now we should see how much this iron by using this hydrophore, how much we will be able to chelate and provide to the plant. I think it's a very good area you have pointed out, and I think we should definitely take up this one. Yeah, and especially the quantum. We will try to measure the quantum of iron and the types of hydrophores so organisms produce. So if you can make a link between high chelation of iron and the hydrophore, if we can make them, we can try to produce these hydrophores in the labs and then ultimately in the industrial base, and also can provide to the our farmers so that they will be benefited.
Thank you, sir. Thank you for your information.
Yeah, I'm sorry, there was some disruption. We are traveling.
That way, I think budding researchers should try to work on that and to contribute for our Annadata farmers. Thank you, sir.
Yes. Yeah. Thank you. Thank you. Yeah. Stop here.
Dear participants, now today's our third respected speaker is Dr. Rameshwar Adhikari, sir. Sir is Professor of Central Department of Chemistry and Research Center for Applied Science and Technology, Tribhuvan University. To introduce, sir, I would like to invite Dr. Alok Rai, sir. Over to you, Dr. Alok Rai.
Thank you, Dr. Sir. Am I audible, sir? So, I, Dr. Alok Rai, Head Department of Microbiology. I feel proud and privileged to introduce our today's eminent and dynamic speaker, Dr. Rameshwar Adhikari, sir, Professor of Chemistry at Central Department of Chemistry, Tribhuvan University, currently working in Research Center for Applied Technology, TU. He also served as Executive Director at Recast 2017 to 2020 and as a visiting professor at Rouen University, France, Mahatma Gandhi University, India, Kyoto Institute of Technology, Japan. Dr. Adhikari studied chemistry in Tribhuvan University, Kathmandu, and then graduated from Martin Luther University, Halle, Wittenberg, Germany in 2001. He is a fellow of International Union of Pure and Applied Chemistry and recipient of Alexander Humboldt Fellowship, and also served as Ambassador Science of the Foundation 2012 to 2016. He is a recipient of Polly Chair in International Material Science Prize and Technology Award of Nepal Academy of Science and Technology, that is NAST. Dr. Adhikari is currently a scientific committee member of Polycap International Polymer Characterization Forum, steering committee member of Asian Pacific Conference of Young Scientists, life member of Nepal Chemistry, Chemical Society, and founder president of Nepal Polymer Institute. So he has published more than 170 papers in reputed journals, and he has guided 15 PhD students, and 6 are now completed. This research area includes polymers, biomaterials, nanotechnology, and natural products. So I request Dr. Rameshwar Adhikari, sir, to deliver the lecture.
Thank you, sir. Thank you so much, Professor Alok Rai, sir, for very kind introduction, very nice introduction. Very nice to be here in this platform. Thank you so much, also Ratiram Chaudhary for your speaker in this conference. So can I share my slides?
Yeah, please.
So hopefully you can see my slides in full screen.
It's visible.
So thank you so much. It would have been so nice if we could have made physically, but even though we are making contacts through online conferences, and this is very nice to be here. Thank you so much for organization and for inviting me in this platform. So you can see my title here. I'm sorry that in the abstract I saw that I forgot some words to write. So it is about compostable planes based on polybutylene 88 co-terephthalate of biodegradable polymer and natural fibers. So these are the two girls who mainly contributed to this lecture, to this work that I'm going to present today. So this is the outline of my presentation. I start with a brief introduction to the topic. Everybody knows that health is the primary concern. So biomaterials, medicines, cosmetics are very important. Plastics are important also in transportation, nanotechnology, agriculture and food protection, textile, of course, and sometimes also in environmental and energy issues. So with these advantages, we also see a lot of problems that is coming to our arena, to our cities, even to our villages, that is degrading our environment drastically. So here is an example you can see everywhere. The cities India, in Nepal, in most of the developing countries. So the general motivation is therefore that in plastics, it has become indispensable part of our life. But it would be nice if we have them which degrade and the degradation process can be controlled. And the natural fibers from renewable resources could be reinforcing pillar. And from, if we could do that from waste, if we could make materials, that would be very nice too. And they should not be necessarily high-tech materials. So with this motivation, we started this work. And the experimental section is like this. We start with biodegradable polyester. The structure is shown here. And if we take this material and make scanning force microscopy, these are the micrographs we get. The left one is the lower magnification micrograph with small sphere-like structures. I call it small because the whole scale bar here is 10 micrometer. And if we go into it to any of the spherulites, then we come to this structure. You can see that there are in the so-called spherulites some small worm-like, lamella-like structures. Therefore, this polymer is a kind of semi-crystalline polymers. The morphology is quite similar to that of very low density polyethylene, and so is the application of this material. The advantage is quite biodegradable. And this is also the topic that we are going to talk today. So this is the biodegradable thermoplastic material, has the density given here, has the molecular weight about 150,000 grams per mole, and it has these properties similar to that of low density polyethylene. And these are the advertisement of the company ICIASTSF, which have publicized to make the advertisement of their products. So let us start with the composites. These are the main topics that I'm going to talk today, and very briefly I will also go to some high-tech material if we have the time. So hydrogen is obtained from deacetylation of the chitin, and microcrystalline cellulose was the pillar that was obtained from wheat stock. The processing temperature was 240 degrees Celsius or more, and they were characterized by different spectroscopic methods, electron microscopy, and also XRD. But today I'm going to talk more on electron microscopy results and testing. So let us start with the composites or the brains of hydrogen with biodegradable polymer. It is known that hydrogen is a natural polysaccharide. The structure is given here and comprises an unbranched chain of the units given here. The properties include non-toxicity, biodegradability, biocompatibility, and others, and also have the advantages of easy availability, low cost, and better stability. So we start with this material, and this was synthesized from brown cells using the method given here. Thus, it was first collected, ground to fine powder, then treated with dilute acid so that minerals are removed. Then we treated that with sodium hydroxide solutions in different stages to get chitin and to undergo deep acetylation so that we can get hydrogen. So this is the simplified method we have used, and the result is this. When we start with the brown cells and go up to the hydrogen, and this is the structure we get. This is the scanning electron micrograph of the material that we have prepared. You can see this is the 200 micrometer scale bar. We have the plate-like, flakes-like structures of hydrogen. When we take it and mid-process with the material, we come to the structures is shown here. On the left side, we have the lower magnification of the scanning electron micrographs. On the right side, this is the high magnification. You can see that in the high magnification, the hydrogen flakes have been embedded with a good bonding with the matrix polymer, And these are several layers of the hydrogens, which are several hundred nanometer thick and several microns long. So when we take these materials, with different compositions, different hydrogen, and then we take it to the ground for degradation. What happens? So what do? We take the samples of these dimensions, then bury them below 10 centimeter under the ground. Under given conditions, we water it and we measure the weight loss in different times. So what we see is this is the polymer, 100% polymer. This is the composite with 5%. This is 10%, 20%, and 40%. After 8 weeks, we see that most of the composites disappear inside the soil. Sometimes it was difficult to find the samples which was inserted into the soil. So this is quite old work that we did, and to summarize, we found that the quite well dispersion of the hydrogen was found in the polymer matrix, and up to 60% by weight of the polymer. Hydrogen could be embedded into the polymer matrix. Thermal stability was also good, and it was compostable that I showed from the pictures before. And films were found, however, fit for low load bearing applications. Okay, let us go to another topic. So with those conclusions, we started to do the work with microcrystalline cellulose that was extracted from the wheat stalk. Using this method, we collect the wheat stalk in a preliminary treatment, then moisturization with plastic soda, and the rest of the colored objects were things were bleached with sodium hypochlorite, then steam explosion and strong acid hydrolysis, and then neutralized. Then finally, we have, using this method, the MCC, the microcrystalline cellulose. And this microcrystalline cellulose was then blended with the polymer, biodegradable polymers. There are some electron micrographs. As I already told you before, I will focus mainly today on electron microscope. So this is the composite with the microcrystalline cellulose. I also show here without the treatment and with nanocrystalline cellulose, but I'm not going to discuss more on this topic. So the microcrystalline cellulose had the structures of several microns, several hundred micrometer long filaments with several hundred nanometers thickness. So when we take these materials, how do the corresponding composites with polymers behave mechanically if we take a microcrystalline cellulose or nanocrystalline cellulose, which have still smaller dimensions, and the macroscopically large fibers of the cellulose? What happens? How do they behave mechanically? So these are the results. For macroscopic fillers, these are the typical bamboo floor composites. So we see that in all the composites with the bamboo floor or microcrystalline cellulose, they are from the wheat floor or the nanocrystalline cellulose. So when we add to the composites the filler, then first of all, the elongation at break, the strain at break falls drastically. So from about 800% to more than about 400%. In all the cases, you can see that. Very similar behavior. But still, up to 40% of the material, if we put up to 40% of the filler into it, then still the material is quite flexible. So when we look at deformation structures of the polymer and the blades, then you can see that this is the pure polymer, which was highly flexible with 800% of the strain at break. So why it was like this? We studied the fracture surface of the material after the breakage, after the tensile test. This is the lower magnification, upper one. The lower one is the higher magnification. We see the formation of these fibrillary structures, which of course should have utilized a large amount of energy to hybridization. Therefore, the result is the ductility of the material. So if we go to another composite, for example, with 40% by weight of microcrystalline cellulose, we see very similar structures, hybridization, but the hybridization is localized. For example, in the meat polymer, you have hybridization everywhere, but in the composite, the hybridization is only localized in some places, and in some places no. So where there is no hybridization in the polymer, that could be the place where the cracks could be originated, and that should therefore much lower ductility. Okay, so then what we did is just to understand what happens if we bury the samples into the soil, and if, as we see, if the polymers really degrade and disappear, if they really become the monomers, this was the question we have put in our mind. Then we did some experiment as depicted here. So these are the composites before composting. These are the initial samples. So this is with 10% microcrystalline cellulose. This is with 40%. This is with 60%. So when we bury these samples for 2 months inside the soil, one with 10% has this appearance, with 40% has this appearance, and 60% this one. And after 4 months, the 60% sample will disappear. We could not find it. So this was the results. So we can see it again. Demonstrate that with the filling of the polymer with the microcrystalline cellulose, the degradability of the composites becomes much higher. So the polymer is itself biodegradable, but when we fill it with the filler, biodegradable filler, the biodegradability increases, enhances drastically. So these are the electron micrographs of the fracture surface, just to illustrate what happens after degradation. So the upper one is after 2 months, and this is after 4 months. So if you have questions, you can ask me, but I just want to skip the discussion here. So I want to put my focus here. So after the samples were degraded inside the soil, they were taken out, they were dissolved suitably in the solvent, and they were subjected to gel formation chromatography to find out if there is degradation in, if there is a deterioration in the molecular weight of the samples. So you can see this is the polymer sample, neat one. It was the weight, average molecular weight. So this is about 50K. You just put your eyes here. I just want to guide you in these two samples. This is with a polymer with 40% microcrystalline cellulose. When we look at the molecular weight of this material with the same polymer with 50K molecular weight, we see after 4 months there is a drastic decrease in the molecular weight, average molecular weight from 50 to about 20. So, but still, this is not monomer. This is still a polymer, 18,000 grams per mole. So these things, even if they appear to disappear inside the soil, they make the microplastics. This is the indications. So we are still conducting these experiments to see how long it will take for complete degradation of the polymer to polymer. So just a very brief introduction to one topic that was not planned actually here. This is the work that was done by my colleague, my student, Mr. Thakal, using multi-wall carbon nanotube. We have also published recently the results. I will show you later. With multi-wall carbon nanotube, the question was, perhaps it will be possible to make very good conducting material utilizing this filling with the multi-wall carbon nanotubes. So I show you very quickly some of the results. Left one is the polymer with 2% by weight of multi-wall carbon nanotubes. The right one is with 5% multi-wall carbon nanotubes. So you can see quite good dispersion of the carbon nanotubes. The darker one, by virtue of their higher density, compared to the polymer, they appear darker in the transmission chamber micrographs. So if we do tensile experiments, as we already also did in other cases, we see the stress strain curves with a pure polymer, very high flexibility. If we add different amount of multiple carbon nanotubes, still all the composites are in various amounts very flexible, more than 100% elongation and break. So, and with this material, did some experiment if they are electrically conducting. This was with a sample with 0.5% multiple carbon nanotube. We completed the circuit with this and put a bulb to illuminate here, and it was easily possible to easily make it glow. And with these experiments, we have found that these are quite good, quite good for strain sensing applications. And these applications, for these applications, the work is still undergoing, and we have very recently some research papers by Dhaka and colleagues published in this issue. So with these results, I just want to conclude, make the general conclusion of these results. So it was possible to prepare in facile way the compostable composite films with the biodegradable copolyester. High hybridization was found to be the reason of high ductility of the polymer, and the planes showed less ductile behavior because the hybridization was localized, and no complete degradation of the polymer was found even after burying them inside the soil for 4 months. So there is a clear indication that even after the several months of degradation, even if the samples appear to be disappeared completely, microplastics formation is possible. We are still working on this topic. And we also found that the samples were, I forgot to write it here, that samples could be utilized for strain sensing application. More study is needed for molecular mechanisms of degradation that one of my students is doing right now. So thank you so much. This is, I'd like to work with young colleagues. These are my students. Some of them have already graduated. Thank you so much for your attention, and thank you so much, Ratramji, and all the organizing committee to invite me in this very interesting, nice conference. Thank you. Namaskar.
Hello. Sir. Hello.
Yes, thank you, sir. I finished my lecture.
Sir, can I ask some questions? Very interesting. First of all, very interesting topic just you have presented, sir. And I want to say that, sir, can we work on the degradation of this polymer which you have made? You have just mentioned that after 4 months, the polymer was not degraded completely.
No. Yeah.
Yeah. Sir, so can we target or can we find out some bacteria related to degrade this polymer?
Yes, sir. Yes, sir. I did not mention it here. One of my students is working on this area. Okay. We have also some results, and he is right now screening what kind of bacteria are effectively degrading.
Okay, yeah. That's good.
If we could work together in this topic, that would be nice.
That will be good for you. That's why I'm asking you.
Yeah.
And love it for this one. This is a very interesting one.
That would be great, sir. Please write your email ID here. I will contact you. I will check your email ID.
I have your address here. I will pay you.
Yeah, please. Thank you.
Okay, sir. That's good, sir. Thank you. Thank you, sir.
Thank you, Dr. Rameshwar Adhikari sir and Dr. Anup Rai sir. If anyone is having question, you may ask to Rameshwar Adhikari sir. I believe that no one asked any question. So thank you so much, Dr. Rameshwar Adhikari sir. And dear participants, we are now moving to our to the ending of this technical session 3. And I, Dr. Utpal Dongre, would like to summarize this technical session 3, where the first lecture was delivered by Dr. Kalyani Pusia ma'am. And her lecture was related to the bioinformatics approaches in the science and technology, where ma'am has covered bioinformatics in India, including DPT, nationwide bioinformatics system established by India, growth rate of databases coming from the databases, the different ways of the databases from which the researcher can access the gene information, and those databases include GeneBank, EMBL, and CTBJ. I'm also added biotechnology informatics system networks in India, and also stated the outcome in terms of publication, patents, and trained manpower. I'm also explained in detail about the role of artificial intelligence in diagnosis of the disease and various initiatives taken by the government of India, including MANO. So that was quite a very informative lecture that was delivered by the Kalyani Pusia ma'am. Our second eminent speaker in today's technical session was Dr. Pramod Ramteke sir. Sir has delivered his lecture very informative information about the role of microorganisms in agriculture, plant growth promoting rhizobacteria, and siderophore production stimulates the iron. In Sir's lecture, Sir talked in detail about the ACC deaminase enzyme to break the which acting as the deaminase to break the various nitrogen compounds, and the possible outcomes which are coming from these nitrogen compounds, how they could be utilized for various applications. Sir has talked in detail about this. Sir also talked about the detoxification system in plants or so phytoremediation is also involved in the first topic. So thank you so much, Dr. Pramod Ramteke sir, for being with us. And the third lecture, the last lecture, was delivered by Dr. Rameshwar Adhikari sir. The presentation was delivered by sir was fabulous and very interesting. Actually, all the knowledge in a very good manner and very in detailed knowledge they have shared with us. Rameshwar Adhikari sir, his talk was basically related to the biodegradation of plastic. Thank you for focusing on the problem of plastic, sir, and the way to degradation is by the biological means, including the Chiku sir. Sir has explained this topic in detail with excellent scientific work. So those who have seen the presentation of sir will definitely understand that how in detail sir has worked on these problems and how they tackle all these problems in a broad way to get rid of all the waste, particularly by using the biological things. For example, including the bacteria and those things that sir has told in this lecture in detail. So I, Dr. Utpal Dongre, would like to thank to all eminent speakers, those who have delivered their eminent knowledge in today's session. Apart from this, I would also like to thank Dr. Rahul Kambe sir, Dr. Subhash R. Kulkar sir, and Dr. Alok Rai sir for this session. And on behalf of management member Dr. Ambedkar College, principal Dr. Ambedkar College, and also all my deep and sincere thanks to all the participants and speakers, and I owe special word to Lucia ma'am, Ramteke sir, and Dr. Rameshwar Adhikari sir for being with us. So thank you so much, sir. And with this word, I would like to end this technical session. Now over to Kamre sir, Dr. Rahul Kamre sir, and Dr. Subhash R. Kulkar sir.
I
sir, are you
request,
there?
yes, sir. Thank you so much, Dr. Chairman, sir. I really appreciate your valuable words for our resource persons. In conclusion, your remark was really uh, appreciable, and it inspires our budding research scholars and research students to do some cutting edge research for the global problems that would be helpful for making our environment, making our green earth, planet earth, and for making our system sustainable for the sustainable future. I uh, should proceed for another technical session 4, and I request Dr. Alok Rai sir, Head Department of Microbiology, SK Purval College, Kanti, to please conduct the program of technical session 4, and accordingly also invite the chairman of the sessions, Professor Dr. Dhanraj T. Asram sir, Associate Professor, Department of Chemistry, University of Delhi, for chairing the session. Doctor, over to Dr. Alok Rai sir.
Thank you, sir. Thank you very much. Good afternoon, everyone. Now I will chair this session, and first of all, I want to invite Dr. Subhash Somkwar sir, convener of the conference, to introduce today's eminent speaker, Dr. Mohammad Farooq sir. Over to you, Subhash sir.
Thank you, sir. A very good afternoon to all of you. I'm very much honored to introduce Professor Mohammad Farooq sir before this August gathering on this online platform. Professor Farooq sir is currently working as a professor and head Department of Biochemistry, College of Medicine, University of Health, KSA. He also served as a Vice Dean for Basic Medical Sciences. He served as a chairperson for International Accreditation Committee. He worked as a member of Ethical Committee. He worked as a member of Faculty Board. He is an international. He worked in International Foundation for Science Sweden. He also completed number of eight major projects sponsored by various national and international funding agencies like International Foundation for Sciences, Research Deanship, University of Health, KSA, Department of Science and Technology, India, Council of Science and Technology, India. He supervised total 18 research students, and he published more than 75 research articles in the national and international reputed reviewed journals. He has 7 books and essays and speeches. He authored that reference books. Then he also published 20 book chapters in reputed book publishers like Elsewhere and Speeches. He has He has 12 scientific societies membership, and he is an editor and editorial board member for almost 25 esteemed reputed research journals. He is also act as a reviewer for journals, and he very recently he has completed number of certificate course and diplomas like Babylon Academy Practical Peer Review Course, Diploma in Teaching Skill, Allison, Ireland, Intellectual Property Right, WIPO from Geneva. Professor Dr. Kudus sir obtained his PhD in biochemistry and enzymology in 2007 from AAI DU India. He is a dedicated educator and researcher with 17 plus years of experience in the area of biochemistry and enzymology. He usually used to teach basic and clinical sciences, hybrid curriculum that includes system-based active learning strategies such as EPL and EPL. So there are several role and responsibilities he handled, carried out. The list is long, and because of the short of time, with this brief introductions, I would like to invite the resource persons for this technical sessions. Professor Muhammad Yusuf, please enlighten us with your research works. Thank you so much. Over to Professor Muhammad Yusuf.
Thank you, sir. Thank you so much. Good afternoon to all of the participants and all the organizing committee members. Yes, and I would like to thank the chairman of the session also, and especially for the Professor Ramtekis who facilitated me to join this conference. So thank you, sir, for you also. Yes, today we are going to discuss some of our research. We share the screen.
My illustration is over time.
Can you use it? Okay.
Sorry for the interruption, sir.
Hello.
Hello, sir. You are audible and you are speaking.
Can you? Yes, sir. Can you speak?
Hello. Sir, we are facing some sound problem here. Mohammed, sir?
I will. Yeah, I can hear.
I think some network issue is there.
Okay. I
Can you change some place where the network is okay for you? I think some network problem is there.
Yeah, the mic is okay.
Your side okay? But sir, आपका already
Sir, audible?
Yes. But not answering me.
No, no, no.
Naveen. Hello.
Hello, sir. Hello.
Can you hear?
Sir, your voice is not clearly audible. There is some internet problem on your side, sir.
Let me just
Hello, sir. Your feedback is not working. Hello. Sir. Hello. Hello, sir. Are you in the meeting? Hello.
Oh.
Hello.
Congress, sir.
Yes, sir.
If Mohammed Sharif is not available, then you can ask to Mohammed Firoz. Hello, sir. Can you hear me now? Yes. Hello.
Yes, sir.
Yes, sir. Audible. Okay, just I am going to share my screen.
Yes, sir. Now you are audible. Sir, your screen is visible, sir. Please share your PPT.
Yes. Now you can repeat. Okay, sir. Okay. Okay, sir. Should not now. Yes, sir. It's visible. So thank you so much for inviting me this content event. For this discussion. So we are going today possible and file and how it's going to hide the well important in biodiversity. So the object of this it is to consider what file and is there this by design how it is going to continue by a way economy. Actually, by technical of design item. So that are going to in for retrace our knowledge. I will be at the enzyme that is going to catalyze this system. It is going to basically is going to it is going to product of a specific but the thing is it is usually they are going to have the meaning it is going to destroy the gene there are because in the also which by is going down is going to be high and it is also going to selective with this is the more addition by the enzyme so now the more for the enzyme going to be here 14 under 2030. Well, though a lot of importance are in reality over and it is in market. Now the whole market here about and to be approved by European and not by the district of Japan. So these are the major data difference that all use the to that is capital market. That is that too many times so much intelligent data to even the population growth how data can be there are more reports happening in the growth and this will be process with the pathogen and are going to be by the world and the other strong that the people that in five and so that and I is going to create is country and school to talk particular or that is known as now what the come to us not that to produce the ability the law and how long the shelf life I for getting they have stayed law so that it is more. They are more cost organization and we operate under first and the person is then not to be a class narrow that was type proper experience by the narrow and so what the experience going to be by the might why can be is going to provide because they are adaptive mechanism and now that many mechanisms one is the enzyme which is adopt that all conditions that is as alternative microbe which is going to fight in a particular extreme in winter countries and the mobile the fight the source of the the enzyme is going to act medicine going to be acted from the enzyme. Various logical application and process that support the application not to appear the process to be integrated. So for that, that particular is going to convert into the under that the streets that meant to play an important role of the and that of the bioeconomy. So in other fields of the that is for green biotechnology, red biology, improved ecology. So in all these four major categories of the biotechnology, at least along the while they are going to do a lot. So how it is going to contribute, as you can see, organism which is going to perform that with some and to the economy. So there are many branch of and on biology. There are lots of another biological compounds so that's a large for very important. It is nothing but the art to use the health and the factory development. So that is related to red biotechnology. The third category is the are they products in various industries. These are food-based various. So that will be also important. Yeah, provide their the biology are for their aid in the economy. One is the biotechnology related to biological needs that need that will be of provisional pesticide, biopesticide, maybe fertilizer. It is used in bioremediation that you plant food in biotechnology. So we can use this particular profile treated by this are going to work by this market. Now, there are required specific products or specific process. There are to form a process. Various as we know, long time nowadays there are new tools that is known as surface scripting, and that particular year 2020, Nobel Prize of the Chemistry was given by two scientists. So from this surface technology, to improve product done by genetic or metabolic studies of particular by of one thermosolvent so that the item process. So it is to involve in that particular process that permits. So after many techniques improving micro but there are always debate about the natural creating micro by being anything. But for there are also is going to be in the natural selection process. What you have to get, it is totally then so it is going to stop biodiversity but if you are going to reaction condition, that is also the sustainable to the green that is rule of green communication that can protect our process and the rule. So we micro and macro. So basically, we are going to get the growth which is going to produce specific time. Why only micro stored intelligence to any animal or any type of insect. The reason is that micro conditions are going to survive in many hot conditions because and there are most important thing is that there are easy availability of raw material, even some you can say some waste material from agriculture. Hello.
Sir, excuse me.
Yes.
Sir, you have only 5 minutes left. Please conclude.
Okay. I
Yes.
will do fast.
Okay, sir. Thank you.
So these are the some conditions why they are going to use the microorganism for the production of any mosaic. And the most important thing is that, as you know that one year about 3% microbes are stored in there, even though 97% are unexplored. As you can see here in this slide, if this small body is the known microbes, so this large are unknown microbes. So there's always possibility that if you are going to explore this microbial world, so maybe we are going to get some new microbes or some new products, for example, new enzyme, and that may be useful for our industrial process. So there is always scope that if we are going to use microbes, so we can get some important microbes or some important enzymes or enzymes we can find from this particular microorganism. So what is the chemophile? The organism which is going to survive our life when at very hot condition at which the mesophilic cannot. Okay. So that may be include thermophiles, acidophiles, or metallophiles. These are some examples. So how they are going to survive? These chemophiles, how they are going to survive? Because they have some specific molecular mechanism. For that one is the presence of enzymes. Because the enzymes are going to work at that particular condition, so there will be normal metabolic process and the microbes can survive. This is the one reason for the chemophile which is going to survive at hot condition. Of course, there are too many specific points about the chemophile, but here we are going to discuss only the application part because it's going to contribute biotech economy. So these are the example like chemophile. For example, it may be extreme pH like acidophile, or alkaliphiles, it may be extreme temperature like thermophile or microphile. It may be salinity, for example, hydrophile. It may be other conditions like aerophile, spectrophile, depending upon the environmental condition in which it is going to be survived. And of course, in our Earth, there are mostly all the conditions are present. For example, there are low temperature area also, that is nothing but polar region. If you want to isolate any cyclophile or cyclotolerant microorganism, you are supposed to go area and you can specific microbes and survive. Of course, there are some reasons. For example, this now lakes that have not only low temperature but very high pressure also. So more than 2 hot conditions you can find in particular area. If you want to go for the highest, for example, in volcanoes or at the mountain, then maybe high temperature also. So if you need in the area, of course, you have to isolate from particular data. So these are the some space area depending upon the requirement of your microorganism. You can isolate from that specific area in of course and fortunately we also have variant climatic conditions means if you want to isolate any cyclophiles, thermophiles, or halophiles, or alkaliphiles, or any metal tolerant bacteria, acidophiles. Half that you can isolate from specific region, because in India also we have glaciers, we have salt lakes, we have volcanoes, we have deep sea, hot sea, mountains. Every climatic condition or every hot condition, an area is there. So you go there and you can isolate specific micro according to your requirement. So here some specific insights, some specific profiles, which is most common. For example, thermophile. If you want to isolate thermophile, so of course you have to go for the thermal water area or hot street, and you have to isolate the microbes from the soil or from the water, whatever food is there. You can isolate the microbes in a is going to high, very high temperature, for example, near about up to 22 degrees centigrade, that may be adopted. So they don't thermophile, that is already isolated these thermal aquatics and phytoplankton. So that is going to produce specific enzymes, for example, polymerase, lipase, and of course there are many more. The important thing is that as we know that the discovery was going to be done by discovery of one thermophilic bacteria, that is the Thermus aquaticus, and of course it is going to give specific enzyme that is known as so that in the polymerase bacteria we need high temperature process for any process, denaturation or elongation or any
Sir, I request you to please conclude fast. Sir, please conclude fast.
So, of course, for example, type polymerase enzyme we can isolate from the thermal aquaticus that was isolated. This is the one best example of how thermophile can contribute in our this one was just economy or the production of specific products. So, like that, of course, there are some microphiles which is going to survive at a very low temperature. If any bacteria or enzyme is active, it should be active at a low temperature. So and there are applications also. For example, an application of biodetergent. Biodetergent that is also related to the environment because it's going to reduce the load of the heavy metals and other chemical compounds. So what happens if you want to isolate any cold-active enzyme? So you have to go to a particular area in the cold region, isolate the microbes, and of course that microbe is going to produce cold-active enzyme. One of the example is here you can see that what we did. We isolated one microbe from the Gangotri glaciers from Western Himalaya, and we isolated particular protease in fact by all that was also. So as you can see, the past performance now it is already published. So if there are what is simple detergent and what simple detergent and enzyme, you can see the difference. Now, of course, there are near about 25% this reflection is going to be increased if you are going to use this particular enzyme. The important thing is that if you are going to use cold active enzyme for washing, of course, not need to go for the hot water, so it is going to save energy also. According to one paper published already that you can save more than 2 or 3 major plants, one in the European country if you are going to use cold detergent or you can go for the cold water. So this is also going to save the energy and of course this is environmental friendly also. So of course there are some other examples also. For example, erythrophile, if you are going to access any erythrophile, you will get the enzyme which is active at very low pH, for example, gluconase, carbonate. They have their specific application. For example, one of the anti-cancer agents that was nothing but MMP inhibitor, it was isolated from the erythrophile. So these are some other enzymes which are stable at the acid condition with low pH. This is going to contribute in many other industrial processes also. So like that, alkalophile, you need alkalophile that is going to active at high pH. So of course, you can calculate from the particular soda lake or alkaline region. You can get alkalophilic enzymes, for example, protease amylase, and that is going to apply in various industry, the food industry. These are some example of any alkaline protease. I see this for the detergent industry. You want to go for the step removal and work at the high PS. So that is applicable for the detergent industry. Like that, there are other industry also in the leather industry or in the wastewater treatment also. These alkaline enzymes are going to be used, and that is going to be excluded from the scheme of 5.1. Like that, if there are halophytic microbes, they are going to produce halophytic enzymes, for example, nucleases, amylases, all that one, and it is going to use for the purpose as you can see. One of the enzymes nuclease from the micrococcus species that is nothing but halophile, and it is going to produce flavoring agent 5.180 and that is also going to adopt a high salt concentration near about 3%. So that is important for the particular industry. Now the pair of going to be found in the deeply after going to adopt too much high pressure, means higher pressure can be adopted more than 400 bars. So that paraphilic enzymes are going to isolate, for example, peptidase is going to work at a specific condition and it is going to produce going to produce specific product like trisaccharide that is basically going to produce from the maltodexatride by the spirophilic microbe. Even though it is the trisaccharide, but in the normal condition, of course, if you are going to use normally spirophilic nature, the product formation is going to be. So depending on the need of the, of course, you can go for these are the enzyme that is was going in a wall, but not just yet. These are by we to apply more than 5 example, yes, temperature and salt and all that. So far, that we need
Sir, please conclude. Sir, please conclude.
Yes, so that is the power hemophile. What is the power hemophile? Actually, the hemophile is to adopt a particular climate condition, but it is only in climate condition. If you can want to see like prefer to get it from lake market, आपको macro survive hydrogen or about get enough. It's going to adopt and all. So this is going to play an important role in the detoxification. The person in the more than to water from water. Are going to produce detergent for the food wash. So we need the that at a low temperature and actually high and concentration. So the enzymes are going to adopt the total high pH concentration. How you get it? You can get it from a single microbe, which is nothing but profile and alkaline. These are going to so all three and these are profile at load pressure is going to provide high is going to provide that is going to provide high. So from particular microbe you can see this time that to adopt this condition. This is where all hemophile are participants play important role for just product. So in that 69 that they are code that file that
Sir, we are running out of time. Please conduct, sir.
the they are case for industry case so it will be used
Hello, Mohammed sir. Please conduct.
it will be used as the vegetable source for waste or not. So that's all about the I think I started showing forward slide. So if there are reaction of
Thank you for your nice and wonderful presentation, sir. If anyone is having any question, they may ask to the sir. Thank you so much, sir, sir, for your wonderful talk. I believe that the call is very yes, yes. Nobody is asking. I request Alok Rashter to please interrupt the session and start the next session. Alok Rashter, please.
Thank you, Dr. Mohammed Kadu sir, for your nice lecture. Now, thank you very much. Now, in the series of this lecture, the second lecture is for second lecture, I invite Dr. Sudip Mandal, Joint Secretary of today's eminent speaker, Dr. Iluz, Professor, Department of Physics, University of Sussex, Malaysia. Over to you, Dr. Sudip Mandal.
Thank you, Dr. Alok Rashter. And I feel very privileged and honored to introduce eminent scientist, Professor Dr. Mohammed Idris, a full professor in physics, in magnetic crystallographic field, member of the International Centers of Diffraction Data (ICCD) in metallic and alloy. He is also former president of the MacGrath Alexander von Humboldt alumni. He works in professor of physics at Sussex University, Tunisia. Still now, he has published more than 130 papers and 3 book chapters. And he is also chairman of 5 international conferences. And he is also the reviewer of many international journals of repute. Under his credit, he has guided more than 20 students, PhD students. And he has attended 50 international conferences. So we are highly honored to have you here today, and our participants will definitely benefit by your expertise. So without delay, I request Professor Dr. Mohammed Idris to start with his proceedings. Thank you.
Thank you very much. Thank you very much. I will thank also Professor Kumaji Chodari and Kisvi Ayodana, who invited me to give this lecture in your National Sciences Day celebration in India. I will talk about magnetism, which is different from you, I think. Also, the magnetic caloric effect in perovskite. I give also some history and fundamental, then some material application, and I give also some research about our result of the year in Indonesia. I will start by some history. Magnetic stone steel was discovered in Asia, simultaneously with iron, and then the first application is compass in China. And in the 12th century, compass arrives to Occident, and we have seen navigation in Asia, which together as some development of commerce and fortunes actually is the first industrial revolution. The magnetic interaction was discovered for the first time by Columbus in 1784, and then Australia discovered the artificial magnet in 1825-29, and some electrical motor and protection of iron magnetic field was discovered in and then the Faraday induction phenomena, which give us some industrial protection of electricity, and it is a second industrial revolution. And then in 1988, we have discovered the reaction. Some application of mobility is the Earth, which is discovered by William Peter, which is about 0.2 percent, and the primary electricity is the life of Sierra, which is discovered in 1956 by one Alan Webster. The first users is Manitou-sensitive bacteria, which called Manitou-tactile bacteria, and some animals are particularly flying one. Manitou-tactile bacteria was described for the first time by Richard and in 1975, and then bacteria are able to network along magnetic field line using magnetic crystal, which is called Manitou. We have also some very practical application. The first application is sanitary control of the ready food. The are outside the oxygen molecule that destroy it. And then electron paramagnetic resonance can detect the presence of toxic radicals. The potential application of Manitou are magnetic crystals which are magnetized or aggregate, measuring 35 to 150 nanometers. Manitou osmos are nanometric size from magnetic nanometers with maximum magnetization. They have also important potential application, especially in nanotechnology and in medicine. The second medical application is NMR. NMR is very important to detect the presence of any atom of hydrogen in the body, and with the magnetic application, we can detect the moving of some hydrogen in the ocean, and so we can have some information about the problem in the body of the human. The consequence of the environment system. Here we have an example of the detection of ions obtained in the hormone. Person is subject to external that oriented the oxide. Then the magnetic field is eliminated, and we measure the field produced by the oriented oxide present in the body. This is the experience from Cohen in 1975. This person has eaten a canned green bean. It has the iron oxide in his stomach. And this person has made the welding arc. It has iron oxide in the lung. The future now, magnetic layers structure, which called spintronics. There is energy production also, thermonuclear fusion, tokamaks, and the magnetic caloric effect. In this lecture, I will talk only about the magnetic layers and the magnetic Manitou caloric effect. GM Manitou resistance, which called the GMR in nuclear restriction, from the work of the work of Bert and Grunberg. They have reported the realization of a new kind of GMR devices, and in 2007 they get their Nobel Prize in physics. Nuclear GMR structure, two or more ferromagnetic layers are separated by very thin, about 1 nanometer, which is non-ferromagnetic. Example, iron, chromium, and iron. This part is iron. This part is iron, and between the iron we have chromium, which is non-magnetic. We have the non-ferromagnetic data. At certain thickness, the magnetics between layers becomes easier to control. The strength of electric current between the layers can change by up to 10%. This device is sometimes also called the spin valve. If the structure in GMR mechanism is the space layer, if the space layer is very thin, we have a change in the intensity of one of the magnetic layer can cause the next one to align itself in the same direction. So we have the same direction if they are different direction. If the layer is will increasing the space layer thickness, we have decreasing coupling between the magnetic layers between the two layers. So application of GMR. GMR has begun a new field of electronic spintronic, which is the interaction between electrical conductor and magnetism. The GMR technology is characterized by high sensitivity, good temperature stability, and excellent reliability over a wide density storage for hard disk, magnetoresistive, which is random access memory, and magnetic sensors. Now I will talk about some of our activities. Here we have physical properties study of magnetic, which called magnetic caloric effect. So we have to elaborate our samples using the subject and also solid-state reaction, and then we perform the powder X-ray diffraction, scanning electron microscopy, and transmission electron microscopy, and perform also magnetic measurements as magnetization versus temperature, and magnetization versus upward magnetic field at different temperature, and we perform also electric and electric measurements. The perovskite called with the formula AP03 are offer the possibility of various substitution in the A site and the B site. So we have such formula, and in our laboratory we study especially with the A sites which are lanthanum and the Brazilian manganese or the E site can be substituted by calcium, lanthanum, barium, and also other element, and we substitute manganese by also titanium, iron, chromium. We have performed some series of samples, and we characterize our samples with different methods. Why the use of nanoparticle? Nanoparticle consist of macromolecular materials. Their size varies from 1 to 100 nanometer. This property hold the great promise for application in areas such as microelectronics, electro-optic photocatalysis, and photoelectrocommunication. also particularly attractive because of their large surface area and fast charge transfer properties for photovoltaic and photodegradation of chemical waste and polymer. Generally, we have some generally this perovskite, fungi perovskite have some fundamental property with fundamental research like electrical properties, properties, magnetic properties, and electric properties, and also some application like magnetoresistive sensor, magnetic refrigeration, digital data storage, and the fuel cell for permanent magnet. So one of method to prepare nanoparticle is to solve technique to prepare underground nanoscale because the formation rate is very high that we can introduce into the structure. So the manganese oxide have attract more attention for several investigation. The study of microstructural properties of these materials determine the measure and the rest and the properties of this kind of oxide. This work is a contribution to the study of evolution of microstructural parameter in terms of crystalline size, lattice parameter, thermal and magnetic parameter after the social method. Here we have some structure of this kind of manganese oxide, and the oxygen, the manganese here is in octahedron formed by 6 atoms of oxygen, and which is typical of magnetic. Here we have also the elaboration method, which we also use oxides or also nitrite for to prepare our sample using the soil method. Let's start now for some research. Here is research of an example of refinement from this kind of sample using the program for the X-ray diffraction. Rayfield refinement technique shows that the diffraction peaks correspond of the sample can be indexed, can be all indexed, and in the order of its structure, and there is no impurity was observed in this sample. Here also we have determination of composition of and the homogeneity of our this kind of sample. We use the scanning higher resolution scanning spectral microscopy, and for different elements we have chosen a color, lanthanum, praseodymium, iron, barium, manganese, and oxygen. And when we superpose all the color, we have a white color from this part, so it's means that our sample is homogeneous and there is no impurity in our sample. Here also the after powder X-ray diffraction refinement, we can also have the structure of the sample. Our sample, this is the example of the crystal cluster of our sample, and from this we can also determine or calculate the position for the of all other elements and the occupancy of all the elements in the structure. From this part of X-ray diffraction, we can also calculate the crystalline size and the average lattice strain with Williamson Hall with using this formula. Here we have obtained from X-ray powder diffraction. The crystalline size is about 67 or 66 nanometer for this kind of samples. Now I will talk about the magnetic measurement, which is very important in our study. Here we have magnetization as some function of temperature of this kind of sample with an uplift magnetic field about 0.05 Tesla. We can see that we have a paramagnetic to a ferromagnetic state transition with decreasing the temperature and with increasing the temperature of the sample. The Curie temperature was determined by for this kind of sample, and we can see that when we substitute manganese by cobalt, the Curie temperature decreases from 256 P to 175 P for this kind of sample. What is the magnetocaloric effect? So, which called the MCE. We have used right now content production using a classical technique for compression and relaxation of gas. So they are greenhouse gases that contribute to global warming. These gases are so identified as the main causes of the depletion of the ozone layer. From this, we have some solution, and the solution are the refrigeration without the refrigeration gas and with the low energy conservation.
Within the 5 minutes, so that we are running out of time, sir. It's a request to you.
Okay. So the solution is the magnetic refrigeration is one of the ways that can solve this problem. So what is the magnetocaloric effect? Some materials have heat under applied magnetic field. This phenomenon is called the magnetocaloric effect. Materials providing this effect are called the magnetocaloric materials. By using the magnetocaloric effect, the magnetic point cycle close to temperature is possible. Now, how was the effect discovered? This effect was first postulated theoretically in the work of Langevin in 1905. He predicted that the temperature of paramagnetic materials should have depended on the magnetic field. Magnetocaloric was first observed in an experiment by Weiss and Picard in 1917. Where they could measure the temperature change of nickel upon application of magnetic field. What is the magnetocaloric effect? Is manifested by the changing of thermodynamic state of magnetic materials in an external magnetic field, depending on the condition in which the magnetic field is upper the feather. The change of thermal entropy or adiabatic temperature can be used to a numerical parameter characterizing the magnitude of the magnetocaloric effect. How does the magnetocaloric effect work? With the increase in magnetic field causes the magnetic moment line to parallel to magnetic field, which decreases the magnetic entropy due to the adiabatic condition within the system. The total energy of the system will remain constant and the magnetic. So after that, we can suppress the magnetic field. When we suppress the magnetic field, we have our another state of entropy. What are the magnetocaloric with the magnetocaloric effect? This that include pure elements such as gadolinium or alloys. This is a photo of gadolinium, natural gadolinium. So this kind of natural gadolinium is the reference of material at room temperature, and we have a very temperature about the room temperature. We have also variation of magnetic entropy, which is about 10 joule per kilogram per Kelvin, which have an RCP and higher RCP. The problem is that gadolinium is very close as well as it is easy to oxidize, decreasing which is this performance. So the solution, among the solution is the manganese, which is cheaper materials that are easy to synthesize and have many calorific effect in every temperature. So here is the principle of caloric effect. When we apply magnetic field, so we have heating, so we have magnetization. Then when we suppress the magnetic field, we have cooling, and this is two parts of physical quantity alone to calculate the effect, the caloric effect. This part is from the variation of entropy, and this part is the variation of temperature. This is the formula with magnetic measurement, and this is the formula with using the specific heat measurement, which call it the direct information of this kind of magnetocaloric. So here we have some variation of magnetization as a function of magnetic field at different temperature, and for some series of samples, and this part of magnetization versus updated magnetic field and at different temperature should be done between paramagnetic state and the ferromagnetic state of this our samples. So here we have also performed the variation of the specific heat of as a function of temperature applied the magnetic versus applied magnetic field, and we can see that from this we can reduce the very temperature of our sample, and from this part also we can have the variation of the magnetic entropy as a function of applied magnetic field and the temperature for all the samples. So there is also, which is very important for this kind of samples, which are the criteria of performance, which is used by this formula, is the variation of entropy magnetization of the maximum and the distance between the half of the peak and the half peak of temperature, and the good performance criteria of performance RCP give us a good idea about the using of our samples. So here we have some results also about our results of magnetic properties, which are from two kinds of results: variation of magnetic entropy and the variation of temperature, adiabatic temperature. And we have also the RCP for this kind of sample with different formula. This is the formula for using the magnetic entropy, and this is the formula using adiabatic temperature. Here also some results about the RCP and for some reference gadolinium on the other, and all of the other are for our samples, and we can see that we have sometimes some RCP which is very near close to close to very temperature, and so it's different from those gadolinium lower, so we can improve this RCP using the other method. Here is an example of RCP of other samples also, is deduced from our work. Here also the variation of RCP as well as the variation of magnetic entropy, and then we can see that from this kind of sample we have lower entropy, magnetic entropy, but we have a good RCP. But for the other, this one, we have the difference. We have lower RCP and we have a good value of variation of magnetic entropy. Let's start now to different, another different of magnetic caloric effect, not in science, but in compound with formula contributing to iron 70, which are very interesting for magnetization, which are non-oxygen. Here we have powder X-ray diffraction of refinement of our sample, and we can deduce that there is no impurity in our sample. Here also we have the magnetization versus applied magnetic field versus compressor and applied magnetic field, which is about 0.05 Tesla, and we can see that the Curie temperature is higher than the room temperature, which is about 400 Kelvin. Here we have the structure of this kind of sample, and this is it's not so easy to calculate or determine the structure using the foolproof software. And we have the possibility also to get this structure. Here we have also performance magnetization versus applied magnetic field for this kind of sample. And we have also using this formula, using this result, and using the formula between magnetic entropy and magnetization, we can introduce also and we can calculate also the variation of entropy in this kind of sample. Here we have also a new approach that use the Monte Carlo simulation of to determine the thermal magnetization at different magnetic field, and to determine also the Curie temperature of our sample. And we have also, using the Monte Carlo simulation, we can also calculate the variation of entropy of our sample, and we can see that we have also the same variation of magnetization of applied magnetic field for this kind of sample, as well as the magnetic entropy. What is now the future? Future now is this kind of oxide with magnetic caloric effect. So, as I said, from this kind of sample, we have variation of temperature of half temperature of the peak of magnetic entropy. So the future is how
dear, you know, this is Ratiram. Please try to complete.
using this that we can increase delta T and the layers of in composite, so we can also increase the RCP than using the series of samples at different value of X. So we conclude now. For the manganese oxide, we have some advantages. We have low saturation nanoparticle also are observed, and the no correction and easy to synthesize. It's a low magnetic density. For metallic alloys, we have advantages, high magnetic density, low magnetic saturation, relatively nanoparticle, and we have some corrosion and the mixing, and I did talk about this in this lecture. And both manganese and the metallic alloys are potential magnet to caloric material cooling system based on magnetic refrigeration. I hope to think also our collaborator here from France, Samir Mahmoud from Jordan, Sam is very from Tunisia, Paris,